Dear students, we shall continue the plant breeding for disease resistance. So, this is breeding and development of a cultivating crop plants for disease resistance. So, importance of developing the disease resistance among the crop plants, that also we understood. So, we know that a cultivated crop yield or crop plants, if it is getting affected by any pathogens like bacteria, fungi or any virus, will affect the yield. So, some of the bacterial diseases, can you name some of the plant diseases which can be caused by bacteria? What are those examples? We have the examples which for bacterial diseases. What are the examples? Examples like, yes, black rot of crucifers. Black rot of crucifers, and further examples we'll see with the examples after understanding the some of the crop plants which are developed after this plant breeding program. There we will discuss more. So one of the example for bacterial disease is black rot of crucifers. Then fungal diseases, are many diseases are there. For example, now if you go to the paddy field, if now at present this is harvesting time, so you might have experienced or observed the plants having some of the somewhat a fire, which is after the fire how it will appear a paper or a plant leaf. Like that we can see a scar on the leaf parts and all. So that is also a fungal diseases. So one example, blast of rice, okay, which is caused by fungi. Then many other examples are there for fungi. So examples are brown rust of wheat, red rot of sugar cane, and then late blight of potato. Brown rot of sugar cane. Even the sugar cane plant, if you go if you have nearby, just to see that there will be a red colored mark will be there on the stem as well as on the leaf. So that is also a fungal disease. And problem is that fungal disease can be spread mainly through the air. It spreads through the air. So many of the crop plants or crop yield has been affected. Lot of loss happened for the farmers because of these pathogens. So a plant breeder, will help in developing these cultivars. It will help them to enhance their food production, and also it will reduce the dependence on use of fungicides and bactericides. That's the most important thing as far as our nature is concerned, right? Because when we have discussed in the Green Revolution, I said that they have developed many of the crop varieties developed after applying lot of fungicides, pesticides and all. Suppose those qualities by birth itself, that genetically it has, means this use of chemicals can be reduced. So that is the advantage of this. So by breeding this and development of these cultivars, resistance to diseases will help us to enhance the food production, main thing. Then also it reduces the dependence on fungicides and bactericides. This is what the importance of this. Developing the plant varieties for disease resistance. And a plant breeder must know, before starting this program of plant breeding, a plant breeder must know that what is the causative agent of that disease and how it get transmitted. So he must know causative agent as well as mode of transmission. For example, blast of rice. This is how it will appear, the blast of rice. On the rice rice plant, paddy field, you can see the leaf will be there like this, having these marks on that. And that is actually caused by the fungi. And how it get transmitted? Through air, water, and sometimes through the soil as well as. So those diseases, even you can see some of the white spot on the plant, even you can see some of the leaves are getting ಲಾಂಗ್ ಬೀನ್ಸ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಅಲಸಂಡೆ ಬಳ್ಳಿಯಲ್ಲಿ ನೋಡಿರ್ಬೋದು, ಎಲೆಗಳೆಲ್ಲ ಯೆಲ್ಲೋ ಕಲರ್ ಆಗಿ ಟರ್ನ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಆಮೇಲೆ ಯು ಕ್ಯಾನ್ ಅಬ್ಸರ್ವ್ ಸಮ್ ಆಫ್ ದ ಸ್ಟೆಮ್ ಪಾರ್ಟ್ ಆಲ್ಸೋ ಗೆಟಿಂಗ್ ಡ್ರೈಡ್ ಅಪ್ because chlorophyll content is continuously get absorbed by those fungus, and it will affect the whole plant. So this is what the pathogens cause a lot of demerits on the crop yield. So this problem of this fungus or bacteria or virus, any pathogen, can be overcome by using the two methods of plant breeding program. That is conventional breeding program also there, mutation breeding program also there. So conventional breeding program or conventional breeding method involves just a hybridization method is there and artificial selection method. Okay, so these steps, whatever the steps involved in these conventional breeding methods, are almost equal to the steps what we have discussed in the last class. So this will be identical to those breeding for any other agronomic characters. So we have understood there are 5 steps are there in developing a new variety, right? So those steps are here, say applies here also, but here we will focus on resistant to diseases. So what are the steps can be involved here? The first, there was a germ plasm collection, right? So the germ plasm collection, and main focus is on disease resistance. So those germ plasm collection having disease resistant variety, that is the first focused thing we have to see here, disease resistant. Second step, it was selection of parents, right? So here selection of superior parents having that disease resistant. So second step will be the selection of superior parents with the disease resistant. Okay, then third step, what's the third step will be? The third step will be hybridization of the selected parents. Among these, then hybridization is done among these selected parents. Hybridization among the selected parents. Then next is fourth step. What was the fourth step there? There the fourth step was to collect, you have to evaluate or testing of superior recombinants. Here selection and evaluation of the hybrids, same thing. Of which hybrid here? Selection and evaluation of disease resistant hybrid. Selection and evaluation of disease resistant hybrid. Then the last step is testing and release of new varieties. Testing and release of new disease resistant variety. Testing and release of new disease resistant variety. So this is what the steps involved in the conventional breeding method program for developing the disease resistant. And here the conventional breeding program has limitation. The two limitations for this are, one is the limited number of disease resistant genes. Usually it is difficult to find out the disease resistant genes. Usually the wild varieties crops will have disease resistant will be more among them, but yield will be less. Okay. So one of the limitation for this conventional breeding program is there will be a limited number of disease resistant genes. Second is we have to develop the disease resistant hybrid varieties also. Obtaining a disease resistant variety hybrid also it is also a problem here. So that's why the conventional breeding program is limited by the number of disease resistant genes, and also we can overcome. So if this is the if this conventional breeding program has this limitation, how we can overcome this problem? We can overcome this problem by inducing mutation. what we said already, by genetically we can alter those genetic makeup of that crop plant to see that it has that quality in it within its gene itself, so that no need of applying any artificial fertilizers, pesticides, and so that need not to face any crop loss also. So that is what the mutation means. So this mutation can be done by various means. Okay, so that about the mutation breeding we'll see in the next video. Now some of the examples are there where the crop varieties, some of the crop varieties which are developed as result of this crop varieties resistant to diseases. Not only the crop varieties, specialty is that these crop varieties are resistant to diseases. So what are those list of crop varieties which are developed by this hybridization method, and also which has some of the disease resistant capacity for bacteria, fungi, and virus, etc. So the table is given, it is important. Okay, just mark it as important, this table. So table, in that first you write crop, then crop variety. Okay, second column you can write it as first is crop, second is variety, third is it is resistant to which diseases. Okay, so resistance to diseases, that is a third column, resistance to diseases. Then next is resistance to diseases, next to which causative organism. Okay, which is, what is a causative agent or causative pathogen here. Okay, so the examples we will see in the first wheat. In the wheat crop there is a variety developed named as Himgiri, and this Himgiri, it is a crop variety in wheat plant. It has resistant to leaf and stripe rust hill blunt. It is resistant to diseases like leaf and stripe rust hill blunt. Stripe rust and hill blunt. So these diseases cannot attack on this Himgiri variety of wheat. What is the causative pathogen for this? Is it a bacteria, is it a fungus, or is it a virus? It is a causative agent, it is a fungi. It is a fungal pathogen. So causative pathogen is fungus. Next crop name is Brassica. Brassica means a mustard group, and actually there are many varieties of mustard. Whatever the many when cabbage, all those are the some of the varieties developed from these mustard plants. So the variety name is Pusa swarnim. Very important. Okay, there is for NEET as well as for your board exam also, for 1 mark, or for find out the name variety or give the variety of Brassica which is resistant to these diseases. Like that question used to come. So the name is Pusa swarnim. So this is so another name for this Karan Rai. It has another name. It is Karan Rai. And which disease it can resist? The name of the disease is, which can resist, it is wheat rust. It can resist the disease called as white rust, right? White rust. And causative pathogen. So which pathogen causes these diseases? So this is also a fungal disease. Next crop is cauliflower. Cauliflower, all of you like, right? Gobi manchuri. How it is prepared? Yes. So the variety name is Pusa Sadabahar. Pusa Shubra. Pusa Shubra and Pusa Snowball K1. Pusa Snowball K1. And it can resist the disease. Name of the disease which can resist is black rot. The black rot disease as well as curl blight disease. And this causative agent of this disease is a bacteria. It is caused by a bacteria. It is caused by a bacteria. Then next one is cowpea. Cowpea means a hurulikalu. Cowpea. So its variety name is Pusa Komal. Variety name of this cowpea is Pusa Komal. And it is resistant to which diseases? It is bacterial blight. It can resist a disease named as bacterial blight. So obviously you can make out from which bacteria it will be pathogen it is caused by. It caused by a bacteria. It is caused by a bacteria. Then next example is chili. Next crop name is chili and its variety name is Pusa Shubra over Pusa Swarnim and Pusa Sadabahar. Its variety name is Pusa Sadabahar. Must learn it, okay? You just have some tricks to remember this name so that it will be easy for you to remember this name. So Pusa Sadabahar, it is a resistant diseases for chili mosaic virus. I am writing in a short form. Chili mosaic virus, then tobacco mosaic virus and leaf curl. So all these diseases can be resisted by this variety, and the causative agent is a viral diseases. So these are the list of crop varieties which are developed after this hybridization from the conventional breeding program, or we can say that these are the some crop varieties bred by hybridization and selection for disease resistant varieties. So wheat, the variety name is Himgiri. It can resist the diseases like leaf and stripe rust, Hillblunt disease. Remember this Hillblunt disease which is caused by fungi in wheat. So Himgiri cannot get attacked by this disease at all. Then Brassica, its variety name is Pusa Swarnim. It is resistant to diseases called as white rust disease. Then cauliflower, the variety name is Pusa Subra and Pusa Snowball K1, which can resist the diseases like black rot, curl blight, which is caused due to the bacteria. Then cowpea, its variety name is Pusa Komal, which can resist bacterial blight caused by bacteria. Then chili, its variety name is Pusa Sadabahar, which can resist the disease of chili mosaic virus, then as well as tobacco mosaic virus, as well as it also can resist the disease leaf curl disease caused by the virus. So in the next video we will learn about the mutation breeding as well as plant breeding for insect and pest resistance. Okay, so go through the textbook, read properly, and once again repeat the topic. Remember this name, write this in your notebook neatly and without making any mistakes. Read and remember, use some tricks to remember so that you can get good marks. Let us focus on the topic now, mutation breeding. So plant breeding the alteration on the plant, on the crop varieties against the resistance to pathogens can be achieved either by conventional methods or mutation breeding program. So problem with the conventional breeding program is that there is a limitation in the number of disease resistant genes in the various crop varieties or in the wild varieties. So genetically we have to induce some alteration in the gene so that we can bring the desired alteration in the new variety. So inducing this plant mutation in plants through diverse means, maybe it can be through many methods we can use for inducing mutation. What is mutation? First of all, it is a process by which genetic variations are created. So we are going to create the genetic variation because at the genetic level only we are manipulating this gene. So that's what the plant breeding program definition also follows like that. It's genetic alteration of the crop species in order to get a crop variety which are resistant to diseases, which can get higher yield, etc. So the process by which genetic variations are altered, okay, or created. So genetic variations are created. So we are going to create a new genetic variation. How? By making changes in the base sequence. You know base sequences in DNA, we know that. So in the DNA there are genes are located, right? You know the genes, chromosomes, like that. So actually those quality of any living organism depends upon that gene. So here by mutation we are creating a new genetic variation. How? By changing the base sequence within the genes. So that is a mutation means. Then after doing the mutation, that means after doing some changes in the genetic level, then plant materials are screened. That is, they are screened. We will select the plants with the superior quality after doing this mutation. Then we will identify the desirable genes. That is the next step involved in that. Then next, multiplication of that plant. After this identification of the plant with the desirable genes, then that plant is multiplied with desirable characters. So which character is our focused area? That character, it is made to multiply in a plant by various methods. So apart from this mutation breeding, nowadays we also have some other breeding programs called as soma clonal varieties, developing the soma clonal varieties, and as well as genetic engineering, tissue culture also there. So we are going to study again at the end of this chapter. So this mutation can be achieved by using the chemicals or through the radiation. For example, you have in the human health and diseases, you will study about the cancer. Cancer is caused due to a sudden mutation. So this mutation is a sudden alteration in the genetic level, maybe caused due to any chemicals or through heredity, through also environmental factor, or through any other reasons are there for that. So this both chemicals and radiation. So the gamma radiation can be induced to bring the desirable changes in the base sequences. So that will alter the genetic makeup of an individual. So the chemical mutagens, as well as we can call it as a physical mutagens, gamma radiation. So that both will alter the genetic makeup of an individual. And by this method, we could develop some of the varieties which are able to resist some of the diseases. Two examples let us discuss now in moong bean and also in the bindi. Bindi means bendekai. Here, so one of the example which has developed resistance to yellow mosaic virus, resistance to yellow mosaic virus. Yellow mosaic virus. In which crop variety? You know, in moong bean. Okay, in mung bean, by this mutation breeding program, there is a variety has developed which is resistant to yellow mosaic virus and also poultry mildew. Example 1. So you may ask whether this mutation is successfully done. Is there any examples to support that? Yes, we have one example to support that. There is a resistant to yellow mosaic virus and as well as to the which other diseases apart from that, poultry mildew diseases which caused by virus can be, yes, in mung bean has been developed. That resistance capacity has been developed in that mung bean. And also there is another example. It is resistant to yellow mosaic virus in bindi. Okay, resistant to yellow mosaic virus. In bendekai. Okay, so the resistance to yellow mosaic virus in bindi, that scientific name of this bindi is Abelmoschus esculentus. Abelmoschus esculentus. Abelmoschus esculentus is a scientific name of this bindi in which there is a resistant to yellow mosaic virus has been developed. Okay, then apart from this, so there is a wild variety, several wild varieties of this cultivated species of plants have shown the resistant characters. The resistant characters have shown in the wild varieties. See, the plants which we are growing in our crop field, apart from that, the wild varieties will have shown the disease resistant capacity more. So by the process of this mutation, we could also introduce such resistant genes in the cultivated species too. So that's how we could develop the high yielding varieties in some of the plants. For that example is bindi, that is Abelmoschus esculentus, which has a resistant to yellow mosaic virus as well as poultry, yes, mildew. This is the two examples. So here these examples involves the resistant genes that are in the same crop species. So whatever the example we discussed now, this involves the crop species that has developed a resistant by introducing genes of its own species, not by any other species. So but this transfer of these resistant genes is achieved. So how these resistant genes, for example in this mung bean or in the bindi, we have achieved or we are successfully able to develop or introduce these resistant genes in this cultivated species is by by method is called as sexual hybridization. That achieved by sexual hybridization, that is between the target plant and the source plant. Okay, so the technique followed here, it is the sexual hybridization between the target plant as well as with the source plant. Then the next method is followed by the selection. This is how we could develop some of the resistant genes through mutation breeding program in some of the crop varieties. Then next about the plant breeding for insect and pest resistant. Yes, by insect and pest. So here this is we are going to develop. How can we develop the plant, or what are the examples for the plant which has developed the resistance for the insect and pest? when we talk about the insect and pest, the pest and insects are the major pathogens which affects the plant population in a large scale. Okay, they destruct the plant in a very crucial manner by which the farmers has to face a lot of loss from their crop yield. So what are the reasons? So insect resistant for this crop in the crop is mainly due to the reasons like the resistance in a crop plant against this insect and pest may be due to it has reason, maybe morphological reasons are there, biochemical reasons are there, and as well as physiological reasons also there. So these crop which are resistant to, see, some of the crops will not attract any insects, and some of the crop plants or even some of the garden plants, if you look also, will easily attract the insects and easily get affected by this insect and pest, and some of the crop will not eaten by any of the insect or pest. The reason is because of their biochemical reason, or maybe due to their hairy leaves, or due to some of the thorns. So that's why, see, cows will not graze on all the plants. For example, we use ecca plant. You know ecca plant. So that is a calotropis. So ekki gida anta ಹೇಳ್ತಾರೆ ಕನ್ನಡದಲ್ಲಿ. So this calotropis plant cannot be grazed by the cow at all. Reason is that it has some of the chemical in it. Okay, that's a biochemical reason. So that will cause a cardiac arrest. Then some of the plants cannot be eaten or attacked by any insect or pest because of the hairy leaves. Okay, and some of the insect, you know that they will attract the insects for pollination by producing some nectar or by producing a smell, all those. So the reasons for attracting or resisting the pest or in the insects are reason, maybe in biochemical reason will be there, morphological reason will be there, and physiological reasons will be there. So by altering these genes at this level, we could develop some of the resistance in some of the plants. For example, resistance to jacids in cotton. Okay, cotton plant. In the cotton plant, resistance to jacids is an insect. It has been developed. Okay, it is developed by this, and as well as in wheat, solid stem will not attract any bollworms. Okay, solid stem means stem with a very solid nature. It will not attract by stem sawfly, and as well as smooth leaved and nectarless leaves also do not attract any bollworms. Then as well as in maize, if you take the example of maize, the maize stem borers are there. That means the maize stem will be eaten by some of the insects or pests. So some of the chemical content like high aspartic acid, then as well as sugar content, then low nitrogen and sugar content in maize. If this alteration is if it is done and it is proven already. So in maize, this high aspartic acid, then low sugar content as well as the low nitrogen content has developed the resistance to maize stem borers. Okay, so these are some example to support how the insect and pest can be resisted by having some of the biochemical reason, or maybe through the morphological reasons, or through some of the physiological reasons. So this by breeding method, there is also lot of changes, or where crop varieties are developed in the crop by this hybridization method and selection for insect and pest resistance. So there is a table given in the page number 175, table 9.2. Just look at that table. Okay, so that table you have to write and learn properly.